So I'm going to have three large topics. Hopefully I can cover it in 20 minutes. And I really want to talk about bile acids. I think it's great that at the viral hepatitis meeting we now actually have to talk about bile acids. But really because of NASH, we really have to talk about bile acids. So I'm going to talk about bile acids in cholestasis. I'm going to talk about it in NAFLD, and I'm going to mix some of the different targets. But I really have to talk about the landscape with the different abbreviations with FXR, TGR5, ASBT, and NCCP. You probably know the latter because that is the entry site for Hep B. And in my world, this is fabulous. My PhD work was on Hep B, and so I work on bile acids, so I really need to know very little if I know about NCCP and bile acids and Hep B. So here's really, I think, what you need to know about bile acids. I think they're phenomenal molecules. They've been around for hundreds of millions of years. And they're unique in the sense that they're precious. They need to stay in the body. And they look and act just like hormones. I mean, they come from cholesterol. There's 14 enzymes that you need to make the bile acids. The two main bile acids in us are Colic Acid, the trihydroxylated form here. It's usually amidated here with either a glycine or a taurine. Chenodeoxycholic acid is the other one. It's a dihydroxylated one. These are primarily synthesized. These are called the primary bile acids. The recirculation process is amazing. It goes through 4 different places: the liver, the intestinal milieu, the portal vein, the microcirculature of the liver, the hepatocyte milieu. And if you radiolabel a bile acid, it's 95% of these are there at the end of the day. So you go through a variety of different milieu, and yet it's still there. So these are kind of precious molecules to recirculate. And there are specific transporters, but very little redundancy in the system. So the exporter for bile acids on the canalicular surface of hepatocytes is one molecule and one molecule only. And similar ideas pervade. So if you want to target certain processes, you have actually one target. So that's really quite helpful. Each bile acid circulates however many times you eat. So roughly 8 or 10 times a day. There are luminal bacterial modifications. I'll talk about this. And what do we talk about the biology of bile acids? There's feedback regulation of bile acids. It regulates bile flow. It's involved in the absorption of fats and fat-soluble vitamins, predominantly these. And in fact, it's so important that in cholestatic infants, many of these are so vitamin D deficient they develop rickets within the first couple months of life, or fractures. So it's really quite important in our world. And as Arun alluded to, many of the pathways that he showed are really also bile acid regulated. So what happens? Other bile acids, and I'll get through this in the talk. The bacteria modify the bile acids, and you can take this trihydroxylated bile acid, and with bacterial salt hydrolysis, get rid of the 7-alpha hydroxylase, and you get this bile acid, deoxycholic acid. And the other one that we know clinically is the one that's approved before the last one that was approved for liver diseases, ursodeoxycholic acid, something we don't make because we can't epimerize at the 7 site, but bears can make it, and other organisms. But it's a very hydrophilic bile acid. I show these because we lump together the term bile acids as, oh, bile acids do this and bile acids do that. Well, each one is really a different hormonal molecule. We don't mix up testosterone and progesterone and say these are, sex steroids and leave it at that. And yet we take all the bile acids and we lump them together and say, oh, this is how they work. So keep that in mind as well. All right. So what are some of the other key components in the primer? On the left-hand side is a really nice picture from an article in Journal of Hepatology this year from Fickert and Wagner, just showing you that the concentrations are not really low. In the liver, in the 50 micromolar, there's no typos here, by the way, so the M's are truly millimolar, not micromolar concentrations. So in bile, the concentrations are in the 20 to 40 millimolar, upwards of hundreds of millimolar. These are gigantic concentrations that are seen in the intestinal tract down to the millimolar range, and in the colon, even with that efficient recirculation process, they're in the hundreds of micromolar. So it's really, the cells are exposed to the bile acids, and in the systemic circulation, it's in the low micromolar level, unless you're cholestatic. So what happens to some of these, and I'll show it on the right in the pie charts, ignore the different abbreviations. I highlighted Colic acid and chenodeoxycholic acid on the first slide, and so in bile it's roughly 70% together, but by the time you get to the feces it's only 4%, and it's a different composition. So across the GI tract, you have different exposures to different bile acids, and different cells see them differentially. And so, depending on your microbiome, this is where there's a whole story of microbiome changes in metabolism. Often some of this is actually bile acid composition related, okay? So certain bacteria metabolize bile acids and change the biliary components. So here's what some people like to see is, well, what do bile acids do? And so here's just a small pie chart, so a small graph I made a couple years ago that there are 6 nuclear receptors that are responsive to bile acids. There's a whole variety of intracellular signals. This is a small list. There's many others. And it's involved also in apoptosis and other pathways. In other words, these molecules, just based on their shape and hydrophobicity and hydrophilicity, can really interact with a number of other proteins, okay? So what I was asked to talk about bile acid-based approaches and what I would consider the revival, the rethinking of bile acids as therapeutics. And here's a figure from one of Michael Trauner's recent papers in hepatology, where we're going to focus on ways where bile acids and the circulations may impact liver disease by thinking about NCCP blockers outside of viral hepatitis, Noroso, which I'll mention, which is, I think, a new molecule that we need to know a little bit more about, FXR ligands that I'll talk a bit more about. And in the gut, something that we've been working on and others is ASBT inhibitors, and ASBT is the sole protein that reclaims bile acids from the distal ileum back into the portal circulation, and ASBT inhibitors as well as bile acid sequestrants. Okay? So in that introduction, we'll talk more about now cholestasis, then I'll talk about NAFLD. Okay? So I want to also mention the abstract 14 from Ghosh that talked also about an FXR activator as well. So what about the FXR targets in the hepatocyte? Here's one where there's actually 7,800 sites in the chromatin where FXR binds. So when people say this is an FXR target, there's another 707,999 to go for. So and then there's layers on this. There's one that's an RNA binding protein that changes the same protein. So it's woven in to liver biology. And here's one that just says that when you activate FXR, you increase the export into the canaliculus, you reduce import from NTCP, and you can increase export back out for renal elimination. Okay? So how important is FXR? Well, if something's important, if you don't have it, and there's no workaround for it, you should be diseased. Well, you're really profoundly diseased if you don't have FXR. Here's a paper that just came out a year ago that people found through exome sequencing of a kid who presented as if they were a PFIC2, but they couldn't get bile acids out of the liver. Really, with exome sequencing, didn't have that disease, but was deficient in FXR. And in the first couple of weeks to months of life, had profound liver failure and liver damage. And once they figured it out in one family, several of these were also transplanted and they did well. The point here is that FXR was discovered in 1999. The first case that was presented with FXR deficiency was 2016. And this was through exome sequencing. So we're going to be updating this whole roster of cholestatic liver diseases and interactions over time. So here's a slide of some of the FXR agonists that are out there. I'm going to mention 2 of these and I'm going to box. So Colic Acid, the natural one, which is EC50 or around 20 micromolar, and obeticholic acid, which has been FDA approved for PBC, has an EC50 for FXR of about 99 nanomolar. There's other ones as well, and some people in the room have been working on them. There's also FXR antagonists. Some of them are actually in the diet. So some of the phytosterols are actually pretty potent FXR antagonists as well. So there's a whole interplay that goes on between diet and FXR as well. Okay, so here we're talking about therapy. So Colic acid, our primary bile acid, is actually an FDA-approved drug over the last couple years, and it's for one group of diseases called bile acid synthesis defects. And in this disease, you have one of those 14 enzymes aberrant, so you keep trying to make more and more bile acids. There's no feedback regulation of that first committed step in bile acid synthesis, so they make all of these really awful bile acids that are not fully functional. They stay within the hepatocyte because they can't be transported out and damage it. So actually you can treat this disease with a bile acid to shut off new synthesis, and it actually restores liver health, avoids death and transplantation, and it's a lifelong cure. So Colic acid, it's really a nice proof of principle that FXR agonism actually does shut off bile acid synthesis in the hepatocyte, and that this is this extreme examples that we learn from in pediatrics that can sort of help with where FXR agonism is going to work for other diseases. And what are those other diseases? Well, here is one of the studies that's published in New England Journal, where Colic Acid, the first FXR agonist that was approved, helps PBC patients, those that they're all on Urso, but they're still not better. And in fact, Alk-Phos, as Arun was talking about, with surrogates that are accepted, a drop in alkaline phosphatase is an accepted response for PBC drugs, and the placebo is in the open circles. The different doses of Colic Acid are in the colored squares, and the alkaline phosphatase levels dropped. So this and others showed that they got better. And some of these patients, though, got very itchy, depending on the dose of their FXR agonist. Okay? Another drug that I think is familiar here is Mercury Dex. So the NCCP inhibitor, well, this, as you all know, it's a pretty potent NCCP inhibitor. It really comes from Hep B. So it's peptides 2 to 47 of pre-S1, inhibits HBV entry, but also what it does in humans is that this is the main way that conjugated bile acids get back into the liver. We didn't know this until this drug existed. And in fact, in human volunteers that received Mercury Dex, they measured serum bile acid levels. They are incredibly high. They're in the 200 micromolar range. Remember that normally they're in the 5 to 10 range, and in cholestasis they're in this range. So this is a very potent way to stop bile acids from getting back to the liver. So I think NCCP inhibitors may be something that we can think about. What's also interesting, and I think that there's some controversy about this, and people here can probably write me on this if I'm wrong, but there are some variants in the Asian populations that make Hep B infection less likely. And so this is, I think, a molecule of interest for liver disease, not just for viral hepatitis. Okay? Oh, and there's a great abstract that's out there about another great NCCP inhibitor that, as you saw yesterday on the review, also may help out NASH. So some of these things are coming full circle. All right, another molecule that I want to mention, Noroso deoxycholic acid. This is Urso. The difference between Urso and Noroso is they're completely different molecules because the side chain is shortened so it can't get amidated. So it doesn't go through that enterohepatic circulation that I showed at the beginning. Michael Trauner ran this great study where they, for Primarysclerosing cholangitis, another disease that really needs drugs, did a 12-week trial, 3 different doses of Noroso, about 40 to 60 in each group, the usual exclusions, and I'll just go through the main data points on this slide. Again, here, alkaline phosphatase is your endpoint, and at 500, 1,000, and 1,500 of Noroso, here's the changes in alkaline phosphatase. If you like it in a different format, a graph goes way down by week 12, and if you stop the drug, it comes back up. GGT also goes down as well, and so because of this, there are now plans for a phase 3 study for Noroso. And we and others are trying to figure out actually how this works. So here's going to be a new bile acid-based drug in our armamentarium. The last couple minutes I want to talk about ASBT inhibitors, which in addition to changing the bile acid composition in the small bowel and colon are also FXR and LXR modulators. I'll go quickly and I may skip a couple slides in the interest of time. There's a couple good reviews if you want to see where ASBT inhibitors may be useful. And just as a reminder, this is the protein that's there just in the terminal ileum and also on the apical surface of cholangiocytes that reclaims bile acids from the lumen of the intestine back into the portal circulation. So inhibitors of ASBT will not allow the bile acids to return to the liver, will reduce your bile acid pool size, and also deliver more bile acids to the colon. Okay? So here are ASBT inhibitors currently in clinical trials. It's a slide from my colleague Paul Dawson, who cloned the ASBT. You can see that there are a number out there. In fact, these drugs have been around for about 30 years because they were first designed to try to reduce cholesterol based on the processes that I mentioned on the first slide. They're in clinical trials for a number of diseases. I made them listed them here on the right-hand side. So let me give you some mouse data, and then I'll give you some human data, and go back to some mouse data and finish up. So here's a mouse model of a disease called PFIC3 where you can't get phospholipids into bile. So you have bile acids that are really damaging cholangiocytes. And you can see a bit here in this mouse. The blue is a pericholangular fibrosis as well. And on drug, it goes away. And so you get both reductions in fibrosis and inflammation by a non-absorbable ASBT inhibitor. In other words, targeting a protein in the intestine, not anything that you're going to absorb, can actually help liver disease. So this is why ASBT inhibitors have an attraction for us in the liver world, and in my opinion, in the metabolic world as well, because of the safety profiles. Okay. In addition, this is a paper from the Lancet this past year for PBC. You can make pruritus a bit better with an ASBT inhibitor in PBC. This is probably the lowest common denominator to ever get a paper into Lancet. I hope I'm not offending anybody in the room, but a 2-week crossover design with 11 in one arm and 10 in the other, somebody knows somebody. So it went in, it seems to improve pruritus, and what you're seeing in the light pink bar is that the Gsk compound improved pruritus compared to baseline. And I'll go quickly through this. And so the last bit is NAFLD. And I want to maybe spend a minute or two that sort of is adding to some of the talks from Zubair, from Arun, and to talk about where I think the NASH world, and as Arun mentioned, this is also something in childhood that is something that is going to worsen over time, and it's all going to be your problem, and I feel personally responsible for not letting it go by when we see these patients. A third of my referrals are for this, and that's around the country. So the prevalence is probably about 10%, as Arun said. And I wanted to mention something that I think may provoke maybe a little controversy is I'm not so convinced that NAFLD and obesity is okay if you don't have fibrosis or progression. And I want to show you some studies that I don't think have gotten as much airplay as they should. This is a fabulous study from the New England Journal article where they took all the recruits from the Israeli army, 60% men, 40% women, so it's not just guys, rock healthy people at about the age of 16, 17, and followed them through a national health service over time. So millions of patients followed, okay? And so I'm going to give you the bottom line here. They followed them upwards of 40 years, okay? And they said, what's your BMI percentile when you came in at the age of 17, and what's your risk of dying a cardiovascular death? Just knowing what your BMI is at age 17. And in fact, most of us would probably think, okay, it's really the ones who are way up there, the BMI of 30 and above, they're at risk. Absolutely not. The take home for me here is you're at higher risk if your BMI is in the 50th percentile. Most of the kids in my son's class are not in the 50th percentile. So you're even at risk at age 17 if you're in the 50th to 75th percentile. So we're watching this train wreck happen in slow motion. The other one is this one where overweight, and again, I'm mixing up overweight and fatty liver disease, where if you're overweight in late adolescence, what's the progression for severe liver disease? And here's a great study, certainly not from the US, because the number who had a BMI over 25 was 3,000, and those with less than 25 was 45,000. We couldn't do this study. But what happens in this study is that if you have a BMI just over 25, you're at a much higher risk of having liver disease than if you don't. And this is our adolescence. So I think that this is something that we need to pay attention to. Okay, I'm going to skip that, and I'll skip the Flint trial. This is the one where the NASH, if you give Colic Acid, you can improve NASH. Arun is the lead of this study from the Flint trial, and basically it used biopsies at the beginning and the end. And the bottom line here is that you can make NASH better with an FXR agonist. I mean, they reached the endpoints, there was less fibrosis, and there was improvements in the NAFLD score on biopsy. So Colic Acid can work there. But through a variety of different preclinical studies, in mouse models, people have tried to reproduce things like this in a variety of models. And the bottom line is that, and I'll show you at the bottom here in the interest of time, is that if you have an FXR antagonism in a mouse model, you can improve NASH. You can do the same thing with an FXR agonist. So there's some real complexity here that we're just scratching the surface. And for this and a variety of reasons that both work, we embarked on a study where we wanted to just reduce FXR function in the ileum and reduce bile acid return to the liver with an ASBT inhibitor. did the mouse model where they put them on a high-fat diet, put them on an ileal bile acid inhibitor, and I'll skip through the rationale for it. It's published last year in Science Translational Medicine, and you can see the results of this. Is that, first of all, in the red line is those on high-fat diet and those on the diet plus ASBT inhibitor. You can just follow red versus blue, and black is just on chow, but it just follows exactly like the high-fat diet with the ASBT inhibitor. They were glucose intolerant, they became glucose tolerant mice. Their livers look like this, and on an ASBT inhibitor they look like this, but the amount of triglyceride that was accreted, and that's the first step in everything else that follows, went down to normal with an ASBT inhibitor. So again, a non-absorbable drug in the intestine helps fix the liver. And what we think is part of the process here is that cholesterol accretion as well is also reduced in these livers, and that also leads to an improvement in liver biology. Okay? Finally, what we found was that the composition of bile acids became more FXR agonistic, and so through an ASBT inhibition, the liver was seeing more FXR agonists naturally from this. This study and some others, Shire has taken their ASBT inhibitor and they're in the midst of an adult trial with NASH. And so we'll figure out this, whether it actually works in humans in the next year or two. Okay? So I'll skip this in the interest of time and get to the summary slide. So I went through 3 parts. Want to spend a bit of time on a bile acid primer because I think it's important for all of us in the liver world. But there really are links between gut, liver, microbes, and select genes that involve bile acids, and we're evolving this information. There are differential effects in the different parts of the body, including fat, which we haven't spoken much about. Individual bile acids have distinct functional properties, as I alluded to. In cholestasis, it's good that there are several arcs for industry or for FXR agonism, ASBT inhibition, Noroso, but also I think what we're learning in this setting with NCCP inhibition may play out as well for certain liver disease. And for NAFLD, I think it's really complex what FXR is doing in the NAFLD world, just because of what I alluded to, at least in the mouse models for agonists and antagonists. We'll wait for the ASBT inhibitor trial. And again, here, just based on the nice poster from yesterday about NCCP inhibition. And I want to thank my colleagues at Emory and thank Ray for inviting me to the meeting and Ray's complete support for everything that we do at Emory.
Thank you. Thank you very much.